Hi, good afternoon. So I'm Harry Karmouty Quintana. I'm assistant professor at the Department of Biochemistry and Molecular Biology, and I also have a cross appointment with the divisions of critical care, pulmonary, and sleep medicine at UT. And I'm co-director of the UT Health Pulmonary Center of Excellence. And as a result of this, we manage a large biobank for lung tissues that we get from our collaborators at the medical center. So my research focuses in several different areas. So primarily we look at fibrosis, which is an aberrant scar formation in that happens in the lung and skin. And here in these images, you can see that there's a lot of collagen, which is blue, which is affecting the normal architecture of these organs. We also study pulmonary hypertension, and here we focus on the mechanisms that lead to smooth muscle cell remodeling, that result in obliteration of the lumen and increased pressures in the pulmonary vasculature. Most recently, we have begun to do research into COVID-19, and in this image, you can see that there's a immunohistochemistry image of a patient who succumbed from COVID-19, and the arrows are pointing at the deposits of hyaluronic acid, which we believe are important in the pathogenesis of COVID-19. And we also have some interest in a new area, which is in stroke and dementia. I'll tell you how we're getting into this area in a moment. And these sort of diseases that we're interested in, the main focus is trying to understand the role of certain mechanisms. And here we focus on two main ones. One of them is looking at aberrant developmental gene expression in fibrotic diseases. And here we have focused on 6-1, which is a developmental transcription factor. And what we found is that it promotes lung and skin fibrosis through various mechanisms. We also do research in alternative polydilation, and this is this happens when RNA binding proteins such as CFM1M25 are depleted, and this results in alternative polydilation and subsequent 3' UTR shortening, allowing transcripts to evade regulation and have increased mRNA translation. So interestingly, in some mice that lacked CF1M25 in smooth muscle cells, they developed pulmonary hypertension sporadically. And we looked at whether we did some behavioral tests in these mice, and they had some learning difficulties. So that's why we're kind of interested in pursuing this angle in terms of stroke and dementia. And none of this work would be possible without the great students that have been in my lab, both current and past students. And these include Scott Cullum, who did a lot of work on lung fibrosis, pulmonary hypertension, alternative polydilation. He was extremely successful, getting 9 papers during the time in my lab, 3 as first author, and he spent a total of 4 years to get his PhD in my lab. Then Corey Wilson, who just got his defended his thesis successfully last week, and he really uncovered the role of 6-1 in lung fibrosis. And so far, he has 4 papers, and we're working on his first author paper at JCI Insight from my lab. He also has an additional one before he joined, and he was with us for 3 years. And then we have also Nancy Waring, who is currently uncovering the role of 6-1 in in skin fibrosis. And she's done a fantastic job at identifying how 6-1 could be involved in transdifferentiation and lipolysis, completely new concepts. And she's expected to finish in 2022, so about 4 years. So that gives a lab average of 3.5 years, and approximately 5 to 6 papers are published by the students in the time they spend in my lab. The students are also successful in getting their own awards. So Corey Wilson got an NIH F30, and Nancy Waring got a very competitive physician training scientist award from the Rheumatology Research Foundation. So I'll stop here, and if you have any questions, please ask me questions or email me, and also feel free to contact my students who hopefully have good things to say.